Hello everybody. Been a while since I rapped at you, but I figured we should sort of get back in the swing of things. And so I'll just really try and continue where we left off and just kind of give a brief recap of where we were last week and kind of extend that into a little bit of a smaller lecture today, and then I'll post some more in-depth lectures as we move along over the course of the week. Okay, so where we left off last week is we were kind of extending beyond Mendel, beyond sort of standard Mendelian genetics. Okay, and so the world, at least according to Mendel, his research was honed in on a very simplified, boiled down version of genetics. And it was important because that really did lay the foundation for our understanding of a lot of inheritance. But it was very simplified, right? So in a lot of cases, Mendel was looking at a single trait, right? And then through his work, he was able to show that this single trait was controlled by a single gene, although he didn't know it as such. And then Mendel was able to deduce the presence of two different alleles for each of these genes, and then depending on the sort of the dominance relationship between these two alleles, ended up with two very clear, distinct phenotypes, right? Okay. So then we extended beyond that a little bit at the end of last week when we started talking about non-Mendelian genetics, okay? And we kept this really in the context of single traits or single genes. And the large key, kind of the big take-home point from last week, was that we were able to see a larger phenotypic variability. And we saw this larger phenotypic variability within a single trait. And we saw some different examples of this. Examples included codominance relationships producing three different phenotype colors in flowers, red, white, and also that intermediate pink. We saw examples of how codominance relationship between different alleles can result in multiple different blood types, type A, type B, and type AB. We then kind of rolled into the mix this idea that there could be multiple alleles for a particular gene. So in the case of blood type, again, there are three different alleles, and so we can end up with one gene with three different alleles, and then based on both the interaction of those alleles and their dominance relationships, we end up with four different possible blood types, okay? So we got four phenotypes for a single gene, okay? So again, this idea of there being larger phenotypic variability. Okay, so we're gonna use this as a jumping off point. We're gonna continue to dive into this and talk about ways that we can get even larger phenotype variability, even larger ranges of phenotype within a particular trait. Okay. So today in this sort of short lecture here, we're going to talk about this next level of variability. And I'm going to bring in some key terms that I didn't actually put on the course Katie reading. So you have to go back into, I believe this is chapter 3. There's a whole lot of stuff about sort of extensions and exceptions to two-gene inheritance. And I'm just going to focus on a couple of key terms here. I'll put those on the open FAQ, the Google Doc, just so you'll know which terms to look for in that reading. Okay, and so this next level of variability is going to be determined by traits that are known as multifactorial traits. So multifactorial traits are very simply traits determined by several factors. Okay, multiple factors, multifactorial traits. Okay, and there are two main types of these multifactorial traits. Okay. And it really depends on sort of what these factors are. Okay, so this first type of multifactorial trait are going to be traits regulated by two or more genes. Okay, so you can start to see how this maybe sort of builds up more variability, right? So last week we talked about variability within a single gene. Varying dominance relationships between alleles, multiple alleles, that can build up greater phenotypic variability. So now imagine the case of we actually now have multiple genes that are interacting to regulate a single trait. Okay, these are multifactorial traits. And so the example I'm going to run through here, kind of a classic multifactorial trait in humans and in animals, is hair color. Okay. So we know of hair color as having a huge range of different phenotypes for hair color. Blonde hair color, brown, black, red, and then a lot of stuff in between too. Right? So this is another important term I'm going to bring up here. So hair color is an example of a continuous trait. Okay, so this is a term I've brought up before. Okay, so it's not that there are any kind of, it's not, hair color is an example of a trait where there aren't these two distinct antagonistic pairs. There's actually a range of phenotypes across the trait. Okay, so that's really kind of what this continuous trait is known as. They're traits with a range of phenotypes. Okay, now typically or commonly, these continuous traits are controlled by multiple genes. That's how you get this sort of huge variability in phenotypes for a particular trait. You can kind of combine the function of a lot of different genes. Okay, where are we at? Okay, so let's think about this hair color a little bit. Let's think about kind of the chemistry of hair color. So hair color is determined by pigments, just like in flowers. Okay, and so there are two main pigments that regulate hair color. One of them is called pheomelanin. The other is eumelanin. Okay, pheomelanin is the pigment that leads to lighter hair color, and eumelanin is the pigment that leads to darker hair color. Okay, so it's really going to be the combination or really the levels of each of these two different pigments that results in this range of hair color phenotypes. Is there high or low pheomelanin versus eumelanin? There are other levels of complexity. It really determines sort of how stable these different pigments are, and all that kind of stuff can really lead to this range of phenotypes within the continuous trait of hair color. Right. So, kind of with these here, it's tempting to think that with these two different phenotypes, or two different pigments rather, that this is regulated by two genes, but that's actually not the case. So we've got pheomelanin and eumelanin. Okay, so it turns out that these pigments, or the levels of these pigments, are controlled by hundreds of genes. Okay, so this is very, very definitionally a multifactorial trait. Okay, so hundreds of different genes, the functions of these genes then combine to regulate the levels of pheomelanin and eumelanin within, say, hair follicle cells, and then consequently in the hair. Okay, and so again, the function of these hundreds of genes then leads to this range of phenotypes. Okay, so I just want to kind of take a step back and ask the question of how is it that hundreds of genes can regulate the levels of these different pigments? How does it happen? And so I think I want to take just a very simplified kind of reminder of the idea that within cells, these genes encode proteins which regulate a series of biochemical reactions. That's really all that's happening in cells. Okay, and so as an example of this, let's take, in this case, eumelanin. Okay, so eumelanin is going to be the final product in a series of enzymatic reactions. Okay, there's going to be some kind of a precursor molecule that can be converted into some kind of an intermediate. We'll call this intermediate number 1, and then through another enzymatic reaction, this intermediate number 1 can be converted into a second intermediate, which then through yet another enzymatic reaction can then finally convert this second intermediate into eumelanin. Okay, this is kind of a simplified version of this one. But remember that at each one of these steps, each of these is an enzymatic reaction. Okay, so there's an enzyme here, we'll call it enzyme 1, regulates this conversion of the precursor to intermediate number 1. Enzyme number 2 regulates or catalyzes the reaction of this conversion of intermediate number 1 into intermediate number 2. And then there's yet another enzyme here that finishes this whole process off and convert intermediate number 2 into eumelanin. Okay, standard sort of boiled down way to look at biochemical enzymatic reactions in cells. Okay, so remember, each of these enzymes is a protein. Okay, and we go back to the central dogma, all these enzymes are encoded by genes. So there's a gene that codes for enzyme 1, there's a gene that codes for enzyme 2, and a gene that codes for enzyme 3. Okay, and these are pretty simplified reactions. So there are genes that code for these enzymes. There are going to be other proteins that help to regulate these enzymes. Those are also coded by other genes, and this is really just one part of these pigments. There's going to be a whole other set of proteins, which are all then encoded by genes that regulate this other pigment as well. So this is just kind of giving you a little bit of a better idea of how hundreds of genes can lead to the regulation of 2 different molecules that can end up, then the combination of the levels of these pigments can lead to this range of phenotypes of hair color. Okay? And so then kind of semi-related to this, this is how we can kind of get phenotypic variability from a biochemical level. Hair color is even more complicated. So this relates to a question that I got from Connor. And it was a good question. So if I've got, I think Connor's question was he primarily has brown hair on his head, but when he grows his beard out, he actually sees red beard hair colors, or red hairs in his beard. And this is very common in men, but also if you can think of sort of, I think everybody has sort of different hair colors, even in their head, and then think of animals as well. And very many animals have lots of different hair colors sort of throughout their body. Think of zebras or tigers, they have stripes, so they've got very different hair colors. So there's this extra level of variability wherein sort of individual hair follicles can themselves, how do I want to state this, produce different hair color. I don't know a whole lot about how this happens, how some hair follicles end up producing brown hair, while some hair follicles end up producing red hair. But I have a hunch that it's probably controlled by the fact that each of these different hair follicles are going to be different levels of expressions of some of these genes, which can then affect the levels of the pigments that are produced by these hair follicle cells, which ends up in the hair itself. Okay, so any kind of differences in any of these hundreds of genes that regulate this trait can result in a different phenotype. Okay, so you can start to see how in the genetics of humans, especially where we've got lots of variability, phenotypic variability, you can start to see how this might occur through these multifactorial traits. Okay, so this is really just looking at the genetic level. Okay, so multifactorial traits, the first type are those that are regulated by 2 or more genes, and you end up with very complex genetic interactions. So the next type of multifactorial trait, we're going to take that even one step farther. Okay, so the second type of multifactorial trait are traits regulated an interaction between genes and the environment. Okay, so we already saw an example of this. Sickle cell anemia. We saw that at low altitude, high oxygen, there was a standard dominance relationship between 2 of the alleles, but as you move to higher elevations and there's lower oxygen, you end up with an incomplete dominance. Not incomplete dominance, with codominance. Okay? So this is not something totally new for us, but the actual kind of real technical term for this is these are known as complex traits. Okay, and there are many, many examples of this. And this is actually, we're going to dive into this a little bit more in terms of how you actually go about determining what portion of this trait is regulated by genes and what portion of this trait is regulated by the environment. These are very important types of research strategies, especially as it concerns human diseases, which have both a genetic component and an environmental component. So you want to be able to tease that out, especially as you want to try and figure out the best way to manage and or treat these kinds of diseases. So let's get to a really kind of simple example of a complex trait. And so our example, we're going to go back to hair color. But we're going to move to a different organism. Instead of humans, we're going to look at fur color, so hair color, in Siamese cats. Okay, I'm not going to draw out a cat. I think that's just going to be beyond my artistic ability for the day. And so we're going to focus in on an enzyme called tyrosinase, or tyrosinase. I don't know if there's a right way to pronounce this enzyme, but this is an enzyme, tyrosinase. Okay, and so this actually catalyzes the conversion. So if we're talking about hair color again, and so this is responsible, or this plays a key role in eumelanin synthesis. Okay, so the precursor for eumelanin is actually tyrosine. You'll recognize tyrosine as an amino acid. So again, one of the things I like about biology is it finds a lot of ways to never have to reinvent the wheel. Okay, so tyrosine can be a building block for proteins as an amino acid, but it can also be used as a sort of starting molecule to make other important biomolecules within the cell. Okay, so in this case, tyrosine is converted into something called dopa, and this is then converted into something called dopa quinone. Okay? And it turns out that tyrosinase, the enzyme, actually regulates both of these steps. Okay? So that's the role of tyrosinase as it comes to regulating hair color. So let's get back to our Siamese cats and our complex trait. Right? So tyrosinase is encoded by the tyrosinase gene. Okay, and as you would expect, there are many different kinds of alleles for of the tyrosinase gene. Okay, so one of the interesting alleles of tyrosinase is one that's actually temperature sensitive. Okay, and this temperature sensitive is often just abbreviated as TS, for temperature sensitive. So this temperature sensitive allele actually encodes for a tyrosinase that has a different sequence. So I believe it's, I'm not quite sure what the genetic change is, if it's a SNP, if it's just a single nucleotide that changes one amino acid, or if there are several amino acids that are different between the normal wild type tyrosinase allele and the temperature sensitive allele. I'm not quite sure the sort of whole amount of genetic variability here. But it turns out that the normal allele you would expect the tyrosinase functions normally at all temperatures. But as the name would suggest, this temperature sensitive allele actually has some temperature sensitivity. Okay, so the TS allele at cooler temps tyrosinase is fully functional, okay? And so it's able to produce lots of eumelanin. Remember, this is our sort of final product, eumelanin, okay? So it's fully functional, and this results in dark fur. So that's what's happening at cooler temps. But at higher temps, warmer temperatures, and just for reference, this ends up being somewhere around, I think there's versions of this illegal in humans. This is around 35 to 37 degrees, 37 degrees Celsius, which is about body temperature. Tyrosinase is non-functional, which means it doesn't make eumelanin at warmer temperatures, which leads to lighter fur. Okay. So this is one way in which the environment can interact with the genetics of an organism to produce trait variability. Right? So our phenotype in this case is going to vary somewhere between dark fur and light fur. Okay? So what I want you to do is I want you to look up a picture of a Siamese cat and sort of going through this kind of description of this temperature-sensitive allele of tyrosinase. I want you to focus in on what regions of the Siamese cat have dark fur and what regions of the Siamese cat have light fur. Okay, and does this actually, does the image that you see, does it actually jibe with the description of this temperature-sensitive allele? Okay, so are the regions of the Siamese cat that are dark, are those regions of the cat that you expect to be a little bit cooler? Because that would be parts where this tyrosinase is fully active and produces dark fur. And then on the Siamese cat, where there's lighter fur, do you think that those are parts of the cat that are actually, you'd expect warmer temperatures, ergo you inactivate tyrosinase and produce lighter fur. Okay, so I want you to look that up on your own and see if that kind of follows. I'll be back at you probably on Thursday with some videos that talk about our next level of gene interaction. We're going to get away from this idea of producing phenotypic variability. We're going to focus more on, again, the inheritance of two genes. We're going to talk about gene linkage, both autosomal linkage and sex linkage. All right.